Hi everyone, my name is Bridget Daughton, and I'm here today to present to you my project on varying nitrogen sources to reduce algae production costs. So I first wanted to broadly answer why biofuels. Biofuels are an excellent form of energy and they can be used as needed. They can also be integrated pretty seamlessly into existing infrastructure. So depending on the way you actually processed the biofuels downstream, you could use them in the car that you're driving today. We're specifically interested in algal biofuels because algae can sequester carbon from the atmosphere that can be converted to biofuels, and they're actually even more efficient at this than trees. They can also produce more oil per acre than other crops such as corn, and for these reasons they're a very desirable alternative to fossil fuels as well as to other biofuels. There are cons to algal biofuels. They are expensive compared to fossil fuels, and they do require a large amount of resources to grow, particularly water, nitrogen, and phosphorus. So the goal of my particular project is to reduce production costs for algal biofuels to make them more competitive with fossil fuels. The question I'm trying to answer is, can algae grow equally well or better using ammonium as a nitrogen source as compared to nitrate? So the reason that we really care about this is because when we deliver 1 kilogram of nitrogen to algae in the form of ammonium chloride, it costs about $200. When we deliver 1 kilogram of nitrogen to algae in the form of sodium nitrate, it costs about $600. So if we were to entirely transition over to using only ammonium chloride, we could cut our nitrogen costs by about a third, or to about a third of what they were previously. So ultimately, ammonium is cheaper than nitrate, which really ties into production costs. The algae strains used in the study are Picochlorum and Chlorella. These are very promising algae strains for algal biofuels because they do have high growth rates and carbon storage, especially compared to other state-of-the-art algae strains used in research. So this is a picture of an environmental photobioreactor, or an EPBR. I use this to grow most of my algae cultures. It is very predictive of which algae strains will perform well in outdoor ponds, specifically because it can actually simulate outdoor conditions. We can perform multiple replicate experiments due to the small scale of an EPBR. So each of these EPBRs can hold about 0.5 liters of algae culture. And they are commercially available, so the parts are very easily replaced. So just to go over the structure of an EPBR, there is a heat jacket which helps to control culture temperature. There's a light source at the top to give light to the algae. A pH probe connects here and goes into the algae culture to actually monitor pH levels. And CO2 is consistently fed into the algae cultures as well as air. So these are the experimental parameters and conditions for my EPBRs. The light-dark cycle is 12-12. The temperature fluctuates between 13 degrees and 30 degrees, as you can see here, and it's consistently fluctuating to 13 degrees at night and then going up to 30 degrees in the middle of the day. And this is consistently happening throughout the course of the experiment. The pH level tops off at 7, and as you can see here, when the culture pH reaches 7, the EPBR sparges more CO2 into the culture, bringing the pH down, and this fluctuation is rapidly occurring throughout the day, as well as throughout the course of the experiment, to keep the pH in a very tight range. The stirring speed is 200 RPM, and these experimental parameters were chosen to mimic the average weather patterns in Mesa, Arizona during the full month of May. For each experiment I am going to show you, there were 4 replicate algae cultures grown in 4 separate EPBRs. So these are the results from the Picochlorum trials with using ammonium as the nitrogen source. So each trial used 4 EPBRs and ammonium as the nitrogen source. And you can see that they're growing very consistently here, and the rate of nitrogen uptake is also very consistent, which is encouraging because it does seem to say that our results are pretty reliable. I would like to draw your eye just briefly here to the initial nitrogen measurements, which we intentionally made very close to prevent any specific algae culture from growing differently due to receiving different amounts of nitrogen. In the picochlorum with ammonium trial 2, we specifically grew up cultures in nitrogen replete conditions, and when they started to level off, we actually diluted all the cultures back and forced them into this nitrogen deplete condition to see exactly how they would grow. And because algae need nitrogen to grow, they actually very quickly leveled off. It's also important to note that for each of these cultures, we do save them and then later quantify the exact amount of lipids and carbohydrates that are in the cultures, because these are the molecules that are produced and stored during the algae growth cycles that can be converted to biofuels. So it's important for us to really know how many lipids are grown and how many carbohydrates are grown during these growth cycles. So this is a graph of the ODs converted to an ash-free dry rate. You can see that this graph includes both of the ammonium trials that I did as well as the one nitrate trial that I did. And when you're looking at the growth in this graph, it seems very similar. When we actually converted the biomass aerial productivity, which is the grams of algae harvested divided by the surface area of the inner part of the canister for the ePBR, we see that these biomass aerial productivities are pretty similar, not just between the ammonium trials, but also between the ammonium trials and the nitrate trials, which is pretty encouraging because it seems to say that picochlorum can grow equally well with nitrate or ammonium as the nitrogen source. So these are the results from the Chlorella sorokiniana trials. So you can see that I grew up in 4 different ePBRs, cultures that used nitrate as the nitrogen source, and then another 4 different ePBRs. The cultures were grown up in ammonium media solution. And as soon as they began leveling off, we did once again dilute back to force them into this nitrogen deplete condition and see how they grew. And it's interesting to note that in both experiments, they did end up growing back up, and this is very likely because algae do have internal stores of nitrogen that they potentially could have been growing off of, but eventually these nitrogen stores do run out and these cultures do level out and stop growing. Another thing to note here is that we did use very close concentrations of nitrogen, again, to start this experiment. Again, for the same reason that we wanted to make sure that our algae cultures were consistently growing without any outside influences from differences in the nitrogen levels. So this is another graph of the Chlorella sorokiniana ODs being converted to ash-free dry weight. And once again, when we're just looking at this graph, it looks like the two different experiments that the algae grew pretty similarly. When we actually look at the biomass aerial productivity, it seems like the nitrate cultures did grow slightly worse than the ammonium cultures, which is interesting to know. We also have some data from experiments conducted in 2015, and it's encouraging because the ePBR experiments conducted in 2015 with nitrate media and Chlorella sorokiniana show very similar biomass aerial productivities. Even the biomass aerial productivities of the experiments done in outdoor ponds are also very similar. So it shows that we're getting very reliable results, even over a 5-year time span, which is really good. So the conclusion from the data we've seen so far is that algae can grow just as well when replacing nitrate with ammonium as the nitrogen source. The future work that I'm planning on doing on this is to compare algae growth using both urea and ammonium. And this is just because urea is an even cheaper source of nitrogen than ammonium, so ultimately it'd be an even more cost-effective solution. With that, I would like to thank you for watching my presentation, and thank you for your time. I hope you enjoyed it.